It talks about, we're going to go through this paper, but to help you understand this paper, I've also given you this NGS technology notes. All right, so if you open up in that tab, okay. Today we're going to do this. We're going to read this paper and see whether we can understand, because I feel that you need to appreciate the strength and the weaknesses of the NGS systems. Before then only you can look at the applications and then how you use NGS in human genetic research. Okay. Now, important notes when we talk about NGS. All right. If we talk about, I'll give you a brief overview of NGS. This is what you should know by now. By now means, I'm assuming you have watched the videos or you have actually made some reading, hopefully. All right. So basically, that means you should be able to answer these questions. You know, what what is sequencing by synthesis? Okay. Now, altogether, there are these systems down here. Okay. Now, as I mentioned, Illumina remains the most popular at the moment. It is sequencing by synthesis. That means it prepares individual, they chop the DNA into small fragments. And from each of the fragment, they put adapters. And then these adapters, you have a chip. All right, you have a chip, like a glass slide, so to speak. So all this will attach because the adapters are complementary to the chip. The chip also has oligonucleotides. It will stay there. Now, the important step here is amplification. This one they amplify to if I'm not mistaken, it's called bridge PCR, right? So it allows the DNA to bend, and then you can actually amplify. Once it's amplified, and then when it's ready to start sequencing, it adds basis by basis. Okay, so that's why it is called sequencing by synthesis. Why does it require amplification? Simply because the technology that they use is that is not sensitive enough to detect single molecules. So that's why they have to get clusters of the same sequence, right? So they do bridge PCR, so that when they do bridge PCRs, that means in one cluster it's just their sequence. You have to imagine a slide where you have all these different clusters. So it's a random placing, right? It's not really a set place where you have all this. So it's a random place where they have a bridge PCR, and then they start amplifying within the cluster. Once you have enough templates for sequencing, then they start the sequencing reaction. And this sequencing reaction, that means they just allow polymerase to synthesize, and they detect synthesis. How do they detect? Later on, we'll see there are two different detection methods. Okay. Now, the Roche, the ROCHE, Roche 454 is also sequencing by synthesis. It is also amplification based. It needs to amplify this, but how does it amplify? Instead of using a bridge like Illumina, they use beads, right? They use beads. So again, these beads have all this oligonucleotides that allows your fragment to bind, only one of your fragment to bind for each bead, and then they do amplification, all right? The difference about this is that this bead is later placed into wells, right? Each individual bead is placed into each well, and then the reaction occurs there. Again, 454 also uses sequencing by synthesis. Okay. Now, the difference between Illumina and 454 is the Illumina uses bridge PCR, the other one uses beads. Okay. Now, API has a different system. It is sequencing by ligation. All right. Now, ABI, when they introduced the system, there's the marketing strength was that to say that their sequencing is accurate. Now, when you do PCRs, you use polymerase, correct? Polymerase are known to make mistakes. They are known to make, like, sometimes as high as 1 in every 200-300 bases, they make mistakes. But in normal sequencing, normally you don't see that because you have a lot of copies being made, right? So at the end of the day, whatever mistakes that are being made are deemed very negligible, right? But the mistakes are still there. Okay. Now, Illumina N454, all right, they have these mistakes. So that means they have to take that into account when they do downstream analysis, right? But ABI 454, sorry, ABI is solid. It's called a solid system. Can't remember. LI is ligation sequencing of what? Can't remember. ABI solid. It boasts to say that it is highly accurate. However, the mechanism is slow, and at the same time, it is also expensive. So that's why, you know, among the three, Illumina sort of wins the race. Okay. Now, if you follow this next-gen sequencing developments in next-generation sequencing, you will come to the term that, you know, we learn next-generation sequencing. Suddenly, there's a second generation, there's a third generation. You know, so when did that come in? So basically, it talks about the need for amplification. When you still require amplification, this is second generation. But when you don't need amplification, now this is like the next generation of next generation, if you know what I mean. All right, regardless of what term you use. Some people, you know, they call it third generation or whatever. But in terms of development, right, Sanger sequencing, the next gen sequencing by synthesis and so on, that one, but required the application step. And then you have the next generation which doesn't require amplification. What is the advantage of sequencing that doesn't require amplification? That means it can sequence from single molecules. Meaning to say, if you can sequence from single molecules, it says that, oh, that means you don't have to amplify. If you don't have to amplify, that means you are not going to make mistakes. All right. Also, because you are sequencing single molecules, you don't need to chop it up. Okay. So again, Illumina 454 are all sequencing technologies that require amplification. It also requires fragmentation, right? But ion torrent, ion torrent still uses amplification, but PacBio, nanopore and onwards are what we call as the third generation. Okay, I'll get back to that one. Okay, so sorry, I was a bit confused just now. I forgot. Ion torrent still uses amplification, but it uses a different method of detection. Illumina and 454 detects fluorescence, but ion torrent uses semiconductor to detect the hydrogen atoms that are being released when synthesis is made. So this enterin is still sequencing by synthesis, but instead of detecting the fluorescence, it detects the hydrogen, right? But and because of that, they are more sensitive. The marketing advantage for that system is that they are able to, I think if I'm not mistaken, the throughput. The term throughput means how much can you sequence in a day, or in 1 hour. So the more sequence you can generate, that's called the higher throughput, right? And then you have PacBio and Nanopore. These are sequencing by detection of single molecule signals, right? Basically, they have improved on detection methods. Remember, Illumina 454 detects fluorescence. So I don't know, whatever system they use to detect fluorescence, it can only detect after amplification. But for these people, the PacBio and Nanopore, PacBio uses semiconductors. Sorry, I'm confused again. PacBio uses nanophotonics, right? They have a well, and then this well, they've got some technology that is called zero wave magnitude or something, But basically, they are able to detect fluorescence by a single molecule. So that's why it doesn't require amplification. So that's PacBio, and then the other one is Nanopore. Nanopore is a special. I kind of like the idea from Nanopore. Nanopore uses pores. If you talk about a membrane, a membrane has molecules, pore molecules. So they use well, I think they design a pore that allows molecules to pass through, and they run a current. Because the pore has a very small opening, whenever a molecule goes through, the current is going to be disrupted. And this disruption can be recorded. And they found that if an A molecule gives a different reading, G molecule gives a different reading, C different reading, and all these molecules give different readings. So by detecting whatever molecule that goes through the pore, they are able to generate sequences. All right. So that seems to be the way forward. Okay. But this is just meant as a summary of all the different methods. Okay. Hopefully, if you have questions, let me know, because we are now going to read the paper. Okay. Now this is a paper titled Next Generation Sequencing Technologies. It is published in 2019, if I'm not mistaken. Yeah, so it's quite recent, so that's why I've actually used it. And because usually we use that book. I don't know whether I've shown you before. Usually I put it inside your spectrum, but I think I've taken it out. It's a whole textbook on NGS technologies in medicine. So I thought, this time around, I will not use that book. So I went around, and lucky enough for you guys, I actually found this this paper. So we can focus on this paper, plus another paper, this one, which I'm in the process of. This is not a paper, this is a collection of papers, but I'm in the process of making notes for you, right? So, right, as I mentioned, this paper is available. I've also given you the notes on it, right? Okay. So you can, I've highlighted what's important, but of course, don't just read the highlighted bits. So, and yes, we'll leave the abstract. It starts off talking about Sanger sequencing, right? I hope as geneticists you should know the development of sequencing methods. Starts off, well, there are a lot of different methods, but what is normally in the textbook is you have the Sanger and then Maxim Gilbert. All right. Sanger is sequencing by synthesis. Maxim Gilbert is by chemical cleavage, but Sanger is by using dideoxy sequencing, right? You can look for videos on it. Then Sanger sequencing was more popular than Maxim Gilbert simply because Maxim Gilbert techniques, when they use chemicals to cut DNA, which means that, you know, these are chemicals that can actually cause cancer. They're carcinogens, right? So you don't want to be playing around with enzymes that can cut your DNA. So as a result, Sanger sequencing becomes the method of choice. So initially they used radioactive material, so you have to use 4 lanes. All right. And then comes cycle sequencing. Cycle sequencing is the one that you have. I think I put up a video on that one, cycle sequencing. And then this is again superseded by automated sequencing. Automated sequencing starts off with a gel system, and now it is running on a capillary system, right? If you don't know what I'm talking about, please find out. All right, these are meant to be as a review. Okay. All right. So, so the gel denaturing polyacrylamide gel and so on. So it's all here. Then talking about, you need to label like you do in the lab where you run a gel. What do you do to label the DNA? You use ethidium bromide. You can use silver nitrate. You can use whatever chemical that binds to the DNA and then allows it to be seen later. But for sequencing, normally, because you're talking about small amounts of DNA, that's why people, they used to use radioactive material, right? 32P or 35S. But later, this is replaced by fluorescence. Okay, and until now, sequencing has always is using fluorescent labels and not radioactive. Although some labs still do. I know for a fact that some labs still do sequencing by using 32P. Also, when we did Sanger sequencing, what we normally do, if you remember the processes of physical mapping and so on, what we needed was we chop the DNA out and then we clone into vectors. We clone into, well, vectors on probably yeast artificial chromosomes. Why do you want to do that? Is because we want to create fragments, a config of clone fragments, and then if it's too big, 2 million is still too big, then you subclone that into manageable clones and then you sequence that. All right. So the limitation of Sanger sequencing is the length, right? Normally in a typical run, even for us, even now when we do sequencing using Sanger sequencing, we're limiting it to about 1,000 or 2,000, right? Rarely we can actually, mostly is 1,000 or less. Right, unless you're doing genome sequencing, okay? So once you generate that, you can go back. Now, remember also at that time, the contigs were there first, then this is followed by sequencing, right? This is very important when you talk about NGS. So the sequences that are generated only needed to be mapped to the clones, and these clones are small, so it's doable. Later on, right, this point, I'm going to emphasize this point, is that you generate short fragments against a background of the whole genome. So that's why it requires a lot of computing ability, Okay, so during this time, I'm going back and forth. During this time, Sanger sequence analysis, when you generate sequence, you only go back to the backbone of maybe the length of the vector of whatever you have cloned before. Okay, so but still, then you arrange them. So this is where you say allow randomly shared small fragments of DNA from a larger original DNA source to be sequenced randomly and computationally overlapped, because you're only talking about a few millions. The fact that the ability to just get one sequence and align and find a patch in a 3 billion base pair genome was still not available then. Okay. So that part of the project is called sequence assembly. And then at that time there was this, I mean, this occurred during my time when I was actually doing my PhD. So we have the Stedon package. The Stedon package is a combination of softwares that allows you to do mapping. Okay, so it says that the combination of Sanger sequencing and the Stedon package for sequence assembly created the ability to sequence genome roughly 10 times as large as originally possible. So they grew from sequencing small clones to larger clones, and then you have this software that allows you to compare between clones. Okay, that was in 1990. Okay, and then there was an improvement with assembly. So when you're talking about assembly, you're talking about, you know, computing ability to find matches. So that even now is a continuously ongoing improvement. Okay, then however, the effort to sequence, assemble, annotate genomes with Sanger still a significant and expensive undertaking, because there's a lot of work needs to be done. You need to clone, you need to subclone, and then it requires a lot of effort, a lot of people, and within one capacity. If you remember, I mentioned in class, I went up and cloned about 2 million bases, and that was an extra huge task for me that I wasn't able to complete. You know, even like 1 or 2 million bases was very difficult at that time. Right? Then comes the technology for massively parallel DNA sequencing. Okay, when you're talking about massively parallel, this is what we're talking about, NGS. All right. So when they talk about NGS, then you start comparing the NGS against normal sequencing. Well, not normal sequencing, against traditional Sanger sequencing. So in Sanger sequencing, you need to amplify first, and then you need to generate fragments, and then you need to label, and then you run this fragment on a sequencing machine, and then you read it. So there's two different processes. But for NGS, they have actually made that into one single process. That's one advantage. Okay. It says primary differentiation is that, unlike Sanger sequencing, massively parallel sequencing approaches do not decouple, enzymatic decouple, do not separate, right? Nucleotide incorporation from sequence data and data acquisition. So, you know, nucleotide incorporation is the one that you cycle sequencing, right? That is nucleotide incorporation, fragmentation, and finally you put into this machine to detect. That is Sanger sequencing. But it says NGS doesn't do that, right? NGS instruments perform both the enzymology and data acquisition in an orchestrated and stepwise fashion, one after the other. But you don't need to have different machines. For Sanger sequencing, first you need a PCR machine. First you need to do extraction, you do PCR machine, and then you have PCR, you run the gel, and then from the run the gel, then you have cycle sequencing again on a PCR machine. Then only you transfer it into a sequencing machine and read. Right, so there are two different machines. But for NGS, no. It says it performs both the enzymology and data acquisition in an orchestrated stepwise fashion, enabling sequence data to be generated for tens of thousands to billions of templates simultaneously. This is another word, simultaneously. Now, when we did Sanger sequencing, we would use gels. That means each lane you can sequence one fragment. So total, I think about 36. And normally you don't use all lanes, so you can, and then you need to replicate. So in the end, you probably can only sequence about 10 or 20 different samples at one time. Nowadays, you have capillaries. All right, capillaries, I think you can sequence now up to 96 samples. Maybe I'm wrong, but yeah. The point is, NGS allows you to use billions of times, right? Because it is only limited by the size of the chip. Okay, so that means this, in terms of generating sequence, this is one superior step. So then going to NGS, it said sequencing by synthesis. That's why I say you need to understand the basis on NGS first, so you know what is this paper is talking about. So sequencing by synthesis, most commercial platforms use massively parallel sequencing are based on the concept of sequencing by synthesis. In essence, this method permits nucleotide incorporation of a variety of enzymes, detection schemes that permit corresponding instrument platform and collect data in lockstep with enzymatic synthesis. Now that's just a fancy word saying that, you do one reaction, you detect. You do one reaction, you detect. So they lock it there, right? So they don't they don't allow. They have that step. They need to control that, all right? Now, in the category of sequencing by synthesis, it's talking about sequencing by so immediately when you start talking, when you're thinking about NGS systems, when you start sequencing by synthesis, which system is not included? The solid, because that one is synthesis by ligation. All right. Sequencing by synthesis talks about Illumina, talks about Roche, talks about Ion Torrent. Right. So these are all sequencing by synthesis. Right. So what are the common concepts of sequencing by synthesis? They need to be fragmented. So they chop into smaller fragments. And then they need to generate sequences, a library of sequences for sequencing. And then this means that you need to put adapters. Remember, when you do sequencing, you have to have primers for synthesis. And then number 3, on-surface template amplification. So regardless of whether it's a chip, as in Illumina, or in beads, as in Roche, so it needs amplification. All right. So these are the 3 main steps that's required. Here it says the on-surface amplification establishes a fixed XY coordinate. So that means to say, you will see if it's a chip, that means where the signal is coming from. So that's one sample. So they are able to actually identify where the sample is. So that's one sample. In the one that uses beads, it is more systematic because they sort of have wells. So now they know where the signals come from. But for Illumina, for example, the software needs to know where the samples are coming from, and they have to decide. Okay. SBS is ultimately limited in its length sequence read. Now, most systems, they generate very short reads. You don't want, because again, the limitation of synthesis. Here it says limitation is actually signal to noise. Signal to noise ratio is a common ratio that is used in many, if you want to gauge whether whatever technique that you have is actually accurate. Now, signal to noise, that means which one is true signal, which signal is actually noise. So what you want is a signal to noise ratio that is very, very high. That means what you get is always signal, only very, very few noises, right? but if your signal to noise is, is equivalent, so that means you're having a very, very bad system. All right. Because if you increase the length, then there's increasing noise. All right. Modification of enzymology. So nucleotide chemistry synthesis as well as more sensitive detectors have yielded improved. So it's saying that, yeah, I mean, newer machines have improved on that, but it can sequence longer, right? So now I think it goes up to about 100, 150, 250 at the moment. Okay, this has impacted how the sequence data are analyzed, as will be described in section of analytical aspects. And then the next section is called detection schema. So how is it detected? This is again different. One is, okay, there are two main detection strategies. One is direct, the other one is indirect. Direct means directly you look at activity of polymerase. Polymerase adds in a nucleotide, and that nucleotide carries a label. So that is directly detection, and that label is fluorescent. Whatever is in comes there and then it's detected. So that means direct fluorescence detection. Or you don't detect nucleotide, but you actually detect the byproduct. When you say you need to detect nucleotide, that means you have to see what nucleotide are being added. A, T, G, and C. A is being given a different color, right, of fluorescence. So that means it detects green, yellow, black, red, or blue and red. So it can read that. But in another system, right, they don't detect. They use just a normal, just the usual basis. They don't need to have different colors. But what they detect is whether or not there's synthesis. Because if there is synthesis, that means it will have a byproduct. So that means instead of reading each nucleotide, it says you put A, you put T, you put G, you put C in sequence, and see which one triggers the production of a byproduct, say pyrophosphate. If you put A, no, nothing happens. B, G, nothing happens. You put T, then you can see pyrophosphate. That means T has been added. So this is indirect detection. Okay. Now, indirect detection is the one that is being used by ion torrent. Remember, they detect the H, they detect the ion, right? So that's why they feel that that one is more sensitive. The first team copies originally from segment. So signal sequencing, you're talking about the direct fluorescence detection by labeling nucleotides. So you need different nucleotides. The second team uses a combination of single type unlabeled nucleotide. So you don't need to label, but the post-reaction detection, you need to detect reaction. Post-reaction is fueled by a chemical byproduct of the nucleotide incorporation reaction. Right? So that means if you have incorporated, if there is a reaction, then you can detect it. This one talks about what happens if you have two bases of the same, so you get higher signal and so on. All right. It says that all instruments using detection, this detection scheme tend to lower accuracy in mononucleotide, but again, this has been improved. Detection of pyrophosphate release, detection of pH. Okay, so Roche, Roche 454 system detects detection of pyrophosphate release during nucleotide incorporation. Pyrophosphate, again, to jog your memory, in the process of replication, you use polymerase, you have triphosphates. And in nucleotide triphosphates, the three phosphates are named alpha, beta, and gamma. All right. The one that is used in the phosphodiester linkage is the alpha. Okay. So beta and gamma will be kicked out, and this is called the pyrophosphate. Right? So one Roche system detects the pyrophosphate, and then secondly, you have the detection of pH change resulting from released hydrogen ions following nucleotide incorporation. That one, this is by the ion torrent. Okay, just me, coffee. All right. So in general, regardless of the type of detection, each platform will process the XY coordinate specific and so on. Okay.